A very good morning, students. Today we are going to concentrate on a very important topic, that is Hardy-Weinberg's equilibrium. A very important topic based on your exam point of view. Now, before we could discuss what is this Hardy-Weinberg law, we have to understand what evolution is. And you know what evolution is. In the last class itself we have studied what is evolution. This evolution is nothing but the change that takes place in an organism, and this change, the change that is taking place in an organism, this change may be any causes, due to any causes. So this change, it is taking place in the organism, and this change is inherited, in the sense this change is being passed from one generation to the another generation. So that is what the evolution is. And we had a beautiful example for evolution is, we are all came from the different organisms, in the sense we came from apes, then monkey, and then human being. So human being has evolved from these of the ancestors. So what happened in the process of our lifestyle? The changes had taken place, and that changes will be passing from one generation to another generation, and that is what evolution is. And these changes are due to certain factors, that is what we studied. Now, how these changes are taken place in an evolution, in an organism? It is because of certain genetic material that or genetic makeup. So variation in the genes. So the change that takes place in the gene. And second one, it is due to certain gene that is getting flown from one species to another, or from one population to another. So that gene flow is also an example for changing of this evolution, changing of particular organisms characteristics, morphological characteristic, phenotypic characteristics, and genetic drift is also a reason for this. And very important, another one is the natural selection is also important for the changes that has taken place in the organisms, and that leads to evolution. Okay, so this change has occurred because of all these various reasons. What are the various reasons? Because of genetic makeup, that means a variation in the gene due to mutation. Okay, so this is due to mutation, that is number one. And second one, it is because of gene that is flowing from one population to another, and because of genetic drift and natural selection. In all this, the changes are taken place, and that is how evolution has occurred in all the organisms. Okay, right. For an example, if you talk about a kind of an example of wolves. In wolf population is an example that we talk about this kind of a changes that is taking place in the evolution. Okay, right. So now in wolf population, the color of the fur has changed from gray to entirely black. So this is kind of an evolution, an example for evolution. So if you talk about wolves population, the fur has changed from gray to black. So what happened in these organisms? The change has taken place. But we cannot judge how this change has taken place. It may be because of a mutation, or maybe because of gene that is flowing, or genetic drift, or some natural selection, and all this process. Okay, so that is what the evolution is. Okay. So today we are concentrating on Hardy-Weinberg's principle. What is that Hardy-Weinberg's principle? Based on Hardy-Weinberg's principle, what Hardy-Weinberg talks about, he talks about the population which is not undergoing evolution. That is what important is. So evolution means a changes that is taking place. But Hardy-Weinberg law focus on a particular population that population is not going to be evolved. That means no change has been taken place in that organism or in that population. So we will talk about now Hardy-Weinberg's equilibrium, and that is the reason we have studied what evolution is now. Now you know what evolution is. Now we are going to study in a one particular population there is no evolution takes place. That means there is no changes taking place. All the organism remain same from generation to generation. That is what Hardy-Weinberg's law. Now Hardy-Weinberg law is valid. That means this law, Hardy-Weinberg law, is valid only for a particular population. That means the population he focused here is the larger population. Very important. So Hardy-Weinberg law has concentrated on the population called larger population, and in this population there is no evolution. That means there is no changes that is taking place in the organism. So that is what the Hardy-Weinberg law. So Hardy-Weinberg law, first of all, it is valid for only the larger population, and two, that population is not undergoing evolution. That means there is no change that occurs in a population. Okay, and this Hardy-Weinberg law states that two things. One is called the allelic frequency. The allelic frequency as well as the genetic frequency or the gene frequency. So it focuses on two things. One is the allelic frequency, another one is the gene frequency. Now you know the frequency is a particular allele that is being increased or remains in that population. So here now we are talking about what is this allelic frequency and what is this gene frequency. Now what Hardy-Weinberg law states that in a larger population there is no evolution takes place. That means there is no changes in a particular organism. So the allelic frequency and the gene frequency will always remain constant. Very important. So this allelic frequency remain constant from generation to generation. That is what important is. It is not undergoing any change. That means the gene which is there in that particular population will never undergo any kind of a changes. Okay, so what do you mean by the gene that is not undergoing any changes? Means it is silently talking to us that in this population there is no, this word talks more in this population. You can find no in this population. So if the gene frequency or the allelic frequency is remaining constant in this larger population, means certain things are not taking place in that population. What is that certain thing? There is no gene flow. So the gene is not getting flown from one population to another population. So there is absence of gene flow. Second one, there is an absence of genetic drift. The gene is not moving, or the allelic frequency is not changed because of this. And in this population there is no mutation. There is no mutation takes place, as well as there is no natural selection. So all these are very, very important in this when you talk about and there is no recombination. What is recombination? Recombination will always takes place during the crossing over. So here there is no crossing over, so there is no recombination takes place. Okay, so now this concept made clear to you. What is Hardy-Weinberg law? The Hardy-Weinberg law is valid only for the larger population, and this population is not undergoing any evolution changes, and the allelic frequency and gene frequency always remain constant in this population from generation to generation, and this remains always constant in a larger population because this is applicable only for the larger population. So since it is remaining constant, there should not be any gene flow, genetic drift, mutation, no natural selection, and no recombination. If these conditions are followed, Hardy-Weinberg law will be applicable. If these conditions are not followed, if gene flow takes place, these changes will take place in allele as well as in the gene. genetic drift, mutation, natural selection, if any one has occurred in this population, means there will be changes in the allelic frequency and gene frequency. Clear? So what is happening here? Nothing is happening, so this population will always remain constant. Clear? Now we'll see some examples for this Hardy-Weinberg's law. So there are the example that we will discuss here is the population of the beetles. Now there are two varieties of beetles are there. One is the dark grey. It is also called as black. The other one is the light grey. Now the gene which is responsible, or the alleles which is responsible for this dark color beetles are capital A, small a, and capital A, small a. These two are the alleles responsible for giving dark color, and small a is responsible for giving light gray. Clear? If you take a population, in the population you will have some 3A alleles which is dominant and this is recessive, and you will have some 7 small alleles. Okay, so you will have the the dominant will be 3 and the small will be 7. Clear? Right. So now this is a gene pool. Now we are going to talk about the allelic frequency and gene frequency. So now the allele frequency. Now in this allelic frequency, the first one we will talk about the frequency of the dominant alleles. Frequency of dominant. Dominant is capital A is dominant and small a is recessive always. Okay, so the first one in allelic frequency, the frequency of the dominant allele, that is we will denote as P. So P represents the dominant allele. Okay, so this is what the A. So A is represented in the form of P. So now that means P is equal to, out of 10 individuals, out of 10 alleles. So how many dominants are there? 3. So it is 0.3. Okay, this is one. Then second one, this is frequency of the dominant allele. Second one, the frequency of recessive allele. Recessive. Now the frequency of recessive allele we call it as small a. So it is represented by Q. So dominant allele is P, A is represented by P. Recessive, small a is represented by Q. So here Q is equal to, how many small a is there? 7 divided by 10. So 0.7. So the formula for allelic frequency is P plus Q is equal to 1. So here P is 0.3 and Q is 0.7 is equal to 1. So see whether in this larger population the allelic frequency remains same between dominant and the recessive, capital A and small a. There is no other gene comes inside, because it is Hardy-Weinberg law follows only in the absence of gene flow, genetic drift, mutation, natural selection, recombination. So here what happens, no allelic change is taking place. So the number of, even you can just take it in the form of ulta. That means the number of small a becomes 3, number of capital A becomes 7. So even it remains same. So in allelic frequency, the formula is P plus Q is equal to 1. So the number of dominant, the number of recessive, together it remains constant. This is what the allelic frequency is. Now the gene frequency is already we studied the allelic frequency formula is P plus Q is equal to 1. This is an equation number 1. Okay, this is allelic frequency. Now gene frequency is, there are two types of allele that we have, capital A and small a. This is a two type of allele. Now here the frequency of different genotype, that is capital A, small a is crossed with another capital A and small a. Okay, so now if we make a Punnett square, capital A, small a, then capital A, small a, so you will have capital A dominant, and one more capital A heterozygous dominant, then here also dominant heterozygous, then homozygous. So we will talk this now. Now here the homozygous means same. So here the two capital A are same, here also the small two A's are similar. So homozygous dominant means it is capital A, so dominant. So it is represented by double P, so it is called P square. Okay, right. So now here next one, heterozygous dominant. So heterozygous dominant, one capital A, one small a. So it is represented by 2 PQ. Clear? Right. So why? Because capital A, small a, you have two things, 2. So it is 2 PQ. It is repeated by into 2. So it is understood. Twice it is there. So 2 PQ. Okay. So it is, so here it is dominant, homozygous dominant is P square and heterozygous dominant is 2 PQ. And here again you will find the homozygous recessive, that is in the form of small a, it is only once, so it is represented by Q, so it is Q square. Okay, so this is what the actual formula for identifying, because tomorrow we are going to discuss some problems, application oriented questions in the same Hardy-Weinberg law. Okay, so in this formula you should remember, in allelic frequency it is P plus Q is equal to 1. The gene frequency homozygous dominant, how it will be calculated by P square, heterozygous dominant calculated by 2 PQ, and the homozygous recessive is Q square. So everything will be calculated to give you a formula called, so this formula is called P square plus Q square is equal to 2 PQ is equal to 1. So this is the formula equation number 2 for gene frequency. Allelic frequency we have here and gene frequency is this. Clear? Right. So now based on this we are just going to give one example, and tomorrow we are going to discuss much very important application oriented questions in the Hardy-Weinbergs law. Okay. So this much you are clear, I suppose. So you know how to calculate the allelic frequency, you know how to calculate the gene frequency. In gene frequency you know the equation for gene frequency, and how to calculate homozygous dominant, heterozygous dominant and homozygous recessive. Clear? Right. So now in this same we will discuss one more concept, that is, here we already know the P is already in allelic frequency I discussed, P is 0.3 and the Q is 0.7. This is what we have discussed already. Okay, so now here we will take up this formula, and now we will discuss what is happening. Now we will discuss this formula. Now before that I just want to tell you some general things. This box that we already studied in botany, like monohybrid hybrid process, is a Punnett square to identify in this population how many dominant genes are there, number 1. In that how many heterozygous are there, how many homozygous dominant, how many homozygous recessive are there, can be easily found out by this kind of a Punnett square in a population. But here in Hardy-Weinberg law we are not talking about a small population. We are talking about a very large population. So in very large population, the number of species, the number of organism will be more. So this Punnett square will not help to calculate how many dominant, recessive, homozygous, heterozygous conditions are there. So Punnett asked the help from the Hardy and Weinberg. This Hardy and Weinberg, they do not know anything about the biological content. So they are mathematicians. So what they have done, they have helped the Punnett to calculate how many dominant recessive are there, heterozygous, homozygous are there, all this by using a formula. So this formula was given by Hardy and Weinberg in order to help Punnett to find out how many types of gene frequencies, gene alleles are there in a particular population by using this formula. But these people do not know anything about the biology, they just know only about the maths. Right. So based on this, now we are going to find out in a large population how many heterozygous dominant are there, how many homozygous dominant are there, how many homozygous recessive are there. Okay, right. So already we know that is P is 0.3 and Q is 0.7. Okay, that is what we got. So that is the formula P plus Q is equal to 1. Right. So now this formula is applicable here, that is P square plus Q square plus 2PQ. Okay, right. So now what is this P square here? This P square is 0.3 square. It's equal to, that is 0.09. So in percentage, if you have it, you will have 9% of P square, means we are talking about capital A is a dominant. So how many, this is actually 9% talks about the frequency of homozygous dominant. Okay, so the homozygous. So in that population, 9% only homozygous dominant genes are there. Okay, right. Second one is Q square. So Q square is already 0.7 square. So you will have 0.49. So 49%. This Q square is small a, recessive. So it is this 49% is the frequency of homozygous recessive. Understand, right? So second one is 2PQ. So that is the heterozygous. Heterozygous we have 2. So that you will have this is 2 within bracket, that is 0.3 and 0.7. So you will have 0.42. So 42%. So this is the heterozygous. So you will have the this is for the heterozygous dominant. Clear? So this is the actual. So how many capital in this population, how many homozygous dominant are there? 9%. How many heterozygous dominant are there? That is 42%. Then how many homozygous recessive are there? That is 49%. Clear? So this is how we are calculating the Hardy-Weinbergs in the principle by using Hardy-Weinberg law, by using Hardy-Weinberg's equilibrium, we are calculating the total number of the alleles which is presented in the large population. Is that clear? Right. So this is very important regarding this. Right. We'll discuss about some 5 different assumptions to the Hardy-Weinberg's law. And we have already discussed in the early part of this session, that is, what are the things that is needed for evolution. And we know the things which we require evolution is, we need genetic drift, we need gene flow, we need mutation, recombination and all this. But what Hardy-Weinberg law talks, in a large population, all these things are absent. There is no gene flow, no gene drift, genetic drift, there is no mutation, no recombination, all this. Okay, so Hardy-Weinberg law says if these things are absent, then in a larger population there is no evolution takes place, and all will have similar genotypic characters. Everything will be same. Everything will be with the capital A. All in this population, everything will be capital A, small a, and small a, small a. In this population, this kind of a gene arrangement, genetic arrangement, gene frequency is not at all changed from one population, one generation to another generation. So there is no involvement of any kind of the organism entering into this population. Okay, right. So now the first exception to Hardy-Weinberg law is, first one is, there is no mutation. If mutation is there taking place in this population, if mutation takes place in this population, definitely what is going to happen is there will be change that takes place in the gene frequency and allele frequency. Okay, so Hardy-Weinberg law states there is no mutation. That means there is no new alleles, no new allelic formations takes place in this. That is important. Okay, this is first assumption. The second one is called random mating. Random mating. Right. So you know what is random mating, but still random mating means there is no preference for a particular genotype. What is this? There is no preference for a particular genotype. So an example, it does not mean that if capital A is there, it should only meet with capital A. It is not that condition. Random mating in the sense the capital A that can even meet with capital A or even with capital A small a and small a. So that is what random mating is. So random mating always talks about, so that is there is no selective mating. Okay, so randomly it means selective mating is what? Only capital A should meet with capital A. That is called selective mating. But here happens random mating. So whenever you talk about random mating, one more point you add to this, there is no selective mating. No selective mating. That point you have to add it. It is not there, but you have to add it. It is understood. Random mating means there should not be any selective mating. So this capital A can meet with any type of the genotypes. So that is what. There is no preference for a particular genotype. Clear? Right. And third one is, the third condition is there is no gene flow. No gene flow. So what do you mean gene flow? That is, if this is the barriers, this is a hill, and here we have some population and here you have some population. So there is no chance of this population to migrate here and this population to migrate here. So everything becomes isolated. So that is what, there is no gene flow at all. Okay, right. So now the fourth one is the Hardy-Weinberg law. It applicable only for the larger population. In small population, we talked about genetic drift. In small population, there will be a chance of a kind of a new allele enters inside. That may be an accident, whatever it is. That is what we entirely discussed in the last section about the genetic drift. Okay, right. So the last one in this, the fifth one is there is no natural selection. There is no natural selection. So what is natural selection? Natural selection means only the fittest will be surviving. But here there is no natural selection, so no alleles are fit to survive. There is no alleles are fit to survive. In the sense, does not mean only capital A will survive. It capital A small a also will survive, small a also will survive. If natural selection is applicable, only the dominant will be expressing, only the dominant will be surviving, the recessive will be not there in the scenario. So this is what the Hardy-Weinberg's law's assumption is. Okay, there is no mutation, no random mating, no gene flow, applicable only for the very large population that should be in the definite size. The population should be, a large population means it should be in the definite size. Definite size. If the size changes, the population, this Hardy-Weinberg law is not applicable. Okay, and this natural selection is not taking place. So what happens is the allele, no allele is fit, all the alleles will enter into another generation. So in next generation, all these three alleles has the chance of entering into another generation like that. Okay, so that is what the Hardy-Weinberg law assumption is. Clear? Right. So today we have discussed about clearly we have discussed about the Hardy-Weinbergs equilibrium, and tomorrow's section we will be discussing about certain application oriented questions in this Hardy-Weinberg law. So some calculations we will see how to calculate the frequency of the dominant allele, recessive, heterozygous dominant, and all this. Clear? Right. So thank you very much.